Hello everybody, welcome back to another video. Today we are going to be learning about enzymes. So what are enzymes? They are proteins that act as catalysts. So the important information here is that they help to speed up metabolism or other chemical reactions in our bodies. So they really help to break down food that comes into our digestive system. So enzymes help break down food that enters the digestive system, and they do this by fitting like a puzzle piece. So that's why we started off by looking at a puzzle piece. It shows that they fit together specifically so that the enzyme is able to break down whatever needs to be broken down. So another way that you can think of this is like Pac-Man. So we have our yellow Pac-Man down here representing the enzyme. And then we have something called the substrate. So the substrate is whatever is getting broken down. Your enzyme and your substrate need to fit together perfectly like a puzzle piece because they need to bind together here at the active site. Okay, so they bind at the active site and then they are able to interact, and that interaction is what happens, or it's what the enzyme does in order to break down the substrate. And that substrate now is in its own individual products. So we went from substrate to products, and now that thing has been broken down. And our enzyme is still able to continue. So if another substrate comes along, it would also just bind and then again get broken down. So our enzyme, our Pac-Man, is able to continue going and continue helping our digestive system in breaking down what needs to be broken down. So something cool about enzymes is that they only can break down the one substrate that they are meant to break down. So in our introduction, we were just looking at, in general, an enzyme is able to break down a substrate. We are actually going to be focusing on lactose as the substrate in the rest of this video, and it's going to help us prepare for an investigation or a lab that we're going to be doing in class together. So starting off with passage 1, it says lactose or milk sugar is a large sugar that can be broken down into glucose and galactose, smaller sugars, by the enzyme lactase. So that's what we're going to be focusing on. These small sugars are then able to diffuse from the small intestine into the circulatory system to be transported throughout the body and used as a source of energy by cells. Okay, so we already know all that. We know that the sugar is going to go into the digestive system. Part of that is the intestine. It is then going to be transferred to the circulatory system so that it can be used throughout the body to make energy and cellular respiration. Okay, so this part is a little bit new. Individuals suffer from lactose intolerance because they cannot break the bond that holds glucose and galactose molecules together. Naturally occurring bacteria present in the human large intestine can digest lactose and use products to carry out their metabolism, that's like their own cellular respiration, and release waste, including carbon dioxide. This causes the symptoms of lactose intolerance, including severe abdominal pain, bloating, and diarrhea. Okay, so let's bring this back to enzymes. So we have our model here that is going to help us to visualize this. So when you drink milk or eat cheese or have any dairy products, that includes this lactose molecule. So down here. So it is a type of sugar, but it is able to be broken down into more simple pieces. So that's what the enzyme lactase is able to do. And the cool thing about enzymes is you almost always know when you're dealing with an enzyme because it will end in ASE, ACE. So lactase is the enzyme that is able to break down lactose. And again, because they have to fit like puzzle pieces, lactase can only break down lactose. It can't break down anything else in the digestive system because it's specific to lactose. So then we know what happens is they bind together at the active site, and then we get our products. So here's our enzyme. We had our substrate. They bind together at the active site to make our products glucose and galactose so that our bodies are able to use that glucose. What we're going to be looking at in the investigation is when someone is lactose intolerant. And when they're lactose intolerant, that's because their body doesn't produce lactose, so they aren't able to break down that substrate. Or sorry, I think I said that backwards. They don't produce lactase, the enzyme, so they can't break down lactose. And so what our passage one started getting into, which we're going to look at in the investigation, is that our bodies, our digestive systems on their own aren't able to break down the lactose using the enzyme, but we actually have bacteria that live in our digestive system. So the bacteria are going to basically eat the lactose, which will break it down, but then they release the output, which causes those symptoms like diarrhea and gas and abdominal pain. So in this case, the bacteria uses lactose. But because, again, it's not being broken down using the enzyme, it releases all these extra byproducts, which leads to the symptoms of lactose intolerance. So we're going to be taking a look at that more in the investigation. But that's why if you or someone you know is lactose intolerant, they aren't able to break down lactose on their own, they might take Lactaid. This is the enzyme lactase in medicine form so that they are able to have that enzyme in their digestive system at the same time as the lactose is in their digestive system, so that if they have something with dairy, that they're able to break it down. Okay, so let's do a quick check for understanding. So we can label this model using two different word banks. We can do the general labels that would apply to any enzyme, and we can also do the specific labels that are going to relate to our investigation. So we're going to do both together. Okay, so for number one, we have the substrate, or in this case, the lactose. For number two, we have the enzyme or the lactase. Number 3, we have the active site. So that's just referring to this area where the substrate and the enzyme will bind so that they can have their interaction, which allows the substrate to break down. And it breaks down into number 4, our products. In this case, glucose and lactose. All right, so to bring it back to our investigation that we're going to do, it says you will investigate the genetics of enzyme production and simulate the mechanisms by which traits are inherited over several generations. Introduction. The human LCT gene codes for a protein, lactase, needed for lactose metabolism. The LCT gene is typically turned off in late childhood when milk dependency declines. It is believed that ancestral human populations, all adults were lactose intolerant. Research suggests that a mutation occurred about 7,500 years ago, resulting in a new allele. Individuals who inherited this new allele continued to express the LCT gene into adulthood and were lactose tolerant throughout their lives. The table, Possible LCT Phenotypes, summarizes the effect of the alleles on the phenotype. So there's a lot of new vocabulary in this passage, and there's going to be a lot of new vocabulary in the lab itself. So make sure you come to class really having a good understanding of enzymes themselves and how they work so that we can apply this to this investigation that has a lot of other new information as well. One main takeaway is going to be the enzyme model that we labeled together and this table right here, that when there's no mutation, there's no lactase produced, and an individual would be considered lactose intolerant. That means not able to break down dairy products, compared to the new LCT phenotype, which is saying this is someone who had the mutation. They were able to produce lactase as an adult and therefore would be considered to be lactose tolerant. Okay, guys, we'll see you in class for this investigation. Bye.